Bonjour à tous et bienvenue au webinaire Québec-Océan. Aujourd'hui, nous sommes très heureux d'accueillir Tessa Page, qui est une chercheure postdoctorale dans le laboratoire de Piero Chialosi. Tessa vient juste d'arriver au Québec. So welcome Tessa. Tessa est une biologiste moléculaire et une aspirante bioinformaticienne qui s'intéresse à l'évolution et au développement des espèces marines, en particulier aux effets des stresseurs abiotiques et biotiques sur les macroalgues et les invertébrés. Pendant son doctorat, elle s'est intéressée principalement aux réponses physiologiques et transcriptomiques des macroalgues classifiées aux facteurs de stress environnementaux, avec une emphase particulière pour les gènes impliqués dans la calcification et les autres processus qui sont soumis à des effets de changement de température ou d'acidification. Actuellement, ces recherches vont intégrer la multiomique et les réponses à différents niveaux chez les invertébrés marins, afin de mieux comprendre la complexité des systèmes biologiques pour pouvoir prévoir les réactions des espèces dans le cadre de scénarios environnementaux futurs. Tessa, this is your turn.
Okay, merci. Okay. Hello, everyone, and thank you for being here. Astrid, thank you for organizing and introducing me. Sorry, my presentation is going to be in English. I am still in the process of learning French, so excuse me. And thank you, Québec-Océan, for having me and doing this event. So today I'm going to present mostly on my PhD research, so parts of my actual thesis that I submitted. And then if I have enough time, I'll probably quickly add in some side projects that I did that I find really exciting. So I thought I'd share them, but they are not published yet. So my thesis was understanding the molecular and physiological responses of tropical coralline algae to a changing ocean. And I did my PhD in Brisbane, Australia, through Griffith University and the Australian Rivers Institute, weirdly enough. And my work was funded primarily by the Australian government and the Research Council there. Okay, good. So I thought I would start off briefly a little bit about myself prior to doing my PhD. So I worked in an undergrad laboratory, actually in analytical and environmental chemistry, and I looked at quantifying metallothionein proteins. And I worked on a method to actually optimize this for ecotoxicology. But I've always wanted to do marine science, and even though I took a little side road there to do ecotox work and more chemistry, I did my master's in primarily marine science, and I did this in Northern California, just north of San Francisco, and also in Plymouth, England, which is where I met Piero. So my master's work looked at 4 different species of porcelain crabs. Porcelain crabs are really cool because they they have a really wide distribution, and some can be confamilial or even congeneric. So I looked at changes in their mineralogy. I also looked at transcriptomic response in response to environmental change. So primarily acidification and warming. And I looked at species across latitude or intertidal gradients, actually. So low intertidal and high intertidal. And we found that different parts of the exoskeleton remain different or keep different concentrations of ions. And we're still working on, and you'll find this throughout your academic career, still working on papers from our master's. Piero will probably harass me about them more later. But we're still working on publishing and finishing off the analysis there. She will definitely find as a student. All right. Then I thought I'd include some pretty pictures as I put on my 20 layers this morning and went out into -3 degrees, which I've never lived in before. But I did my PhD in Brisbane, which is in Queensland in Australia. And that is home to the Great Barrier Reef. And so this is a picture. I guess I have to use this mouse. See it? Yep. This is a picture of one part of the Great Barrier Reef and a very healthy looking part. This is from 2021, actually, of Lady Elliot Island. So that part of the reef is really pretty. So I thought I'd show this beautiful reef before I start talking about how we're damaging the coral reefs, especially the Great Barrier Reef. And this is one of the research stations that I was lucky enough to go to. Lizard Island, and these are our little research boats. And you see our dive tanks, and you see absolutely no dry suits because the water average is about 27 degrees, and that's like a cold summer. So, all right. So I'm going to go really quickly over this because I'm assuming that everyone's very familiar with the Intergovernmental Panel on Climate Change and their graphs, and everyone knows what global warming and ocean acidification are. But for the purpose of my talk, it's good to have some background. So this is IPCC Graph from 2019. And this one I'm showing first, you can see, is looking at global mean surface temperature. You have historical values and then projected future values. So this is low emissions, which is our representative concentration pathway 2.6. And then you have worst case scenario, increased emissions, and probably most likely is this RCP 8.5. So if you follow this projected line, you'll see at least an increase of 2 degrees Celsius by 2100. So with CO2 increasing the atmosphere, not only driving increases in temperature, we're also seeing decreases in surface ocean pH or ocean pH. This is also termed ocean acidification. Again, you can see my mouse, historical values, and then not so bad, and then very bad. So high emissions, worst case scenario by 2100, we could see a decrease in surface ocean pH by around 0.3 units. This is particularly bad, or we think it's particularly bad for calcifying organisms, because as pH decreases, you get a change in the calcium carbonate saturation state. And calcium carbonate is what calcified organisms and mineralized organisms use for their exoskeleton, shell, structure, etc. So when I think about calcified structures, I think about coral reefs. Coral reefs obviously are made up of corals, different species of corals, and other calcifying organisms, and that's what really gives them their structure. So coral reefs are extremely important. They provide economic and ecological functions. So they protect and shelter shorelines from wave action, tropical storms. They're obviously also ecologically very important, and economically, they support the livelihood of millions of people globally. We don't like to do this, but for the sake of presentations, I always include it. If you wanted to put an asset value on the Great Barrier Reef, its total cost would be around $56 billion. So quite a large sum of money. So they're very economically important, tourism, etc. Ecologically, they shelter and are habitat to about 25% of all marine life, which is a very large percent considering how small coral reefs are and how limited their range is. So coral reefs only take up less than 1% of the ocean floor, and they're confined to being around the equator primarily, and also within the first 50 meters of water. And if we look at predicted changes in the ocean, we know that potentially the first 75 meters of water will be the most impacted by warming and acidification. And so if you look at coral reefs worldwide, and I keep forgetting to do this and use my hands, 71% of all coral reefs worldwide have already experienced warming by around 0.25 to 0.75 degrees Celsius increase in temperature, which is quite a lot. You also, like I said, have lots of organisms, not just coral, where's my mouse, that are made up of calcium carbonate. So coral, obviously made up of calcium carbonate, they have hard structures, and also you have other denizens or inhabitants of coral reefs that are really, really important for the structure and the framework of reefs. And those are coralline algae. So coralline algae, or crustose coralline algae, which I will call CCA for the majority of my talk, are calcified red macroalgae that actually cement together and provide structure to coral reefs. They actually calcify within their cell walls, which is an extremely unique process of calcification. It's unlike other species that calcify. So they produce this calcium carbonate in their cell walls, and then that allows them to actually form these crusts and cement together the reef. So when corals might die, CCA can come in and start growing over these coral and stick them back together and keep the structure and the framework of reefs. CCA and corallines in general are really widely distributed. There's about I think over 1,600 different species of corallines. And even within coral reefs, they have really high species diversity. So if you look at this photo, you can probably see about like 6 different species. So all this pink, this branching stuff, this orange stuff, there's some purpley stuff. All of that is CCA. People just look at the reef and they're like, what is this pink rock? But if you touch it and actually look at it under a microscope, you're like, hey, that's awesome. So widely distributed, heaps of species, lots of them within coral reefs. And so, like I said, they're key components, one, because they actually contribute to reef building, and then probably their most interesting task to coral reef scientists is that they're really important for reef resilience because they induce the settlement and metamorphosis of coral larvae. So they can actually attract corals onto the reef, and then the coral babies settle, and then they start to grow on this coralline algae. So CCA and corallines in general are really, really important for reef restoration work that people are starting to do more and more. And they also induce the settlement of things like crown of thorns starfish, abalone, and other mollusks as well, and giant clams, which is quite cool. So CCA, very important to coral reefs. We also know that they are being impacted by changes in their environment, such as warming and such as acidification. So we do know they're sensitive to environmental change. So this is a figure that I made that is in review right now that's looking at different species of CCA across their evolutionary history. So coralline algae have a really interesting evolutionary history in that some species, they all diverged from their common ancestor, and then you have some families diverging earlier on, and some actually being most specious and having more species related to them. So these being the most specious species, Porolithanol codes, this is probably about 20.4 million years ago, whereas you have this family down here that diverged from their common ancestor around 70 million years. So if you look at that, you know that 70 million years ago, we had previous times of increased temperature and increased CO2. So that means the pH of the ocean was super low, the temperature was really hot. So these species, these families potentially actually lived through times of increased temperature and CO2 that are more related to now and even surpass what we're experiencing now, and that surpass 2100 levels. So interesting to think that maybe their evolutionary history of corallines and their environmental history could impact the way that they respond. But we know that despite their evolutionary histories, you see a very wide array of responses from corallines. So this graph over here is just looking at metabolic rate, so photosynthesis, and only in response to increased temperature in combination with decreased pH or elevated PCO2. And most of the points are looking at directly measuring oxygen in the water. The asterisk ones are through PAM fluorometry, which is like this instrument that you can stick on something and it tells you what's happening. And you can relate that to photosynthesis, but it's not as good for measurement. So the red dots are negative effects, and the blue are non-significant effects. Green are significant positive effects. So again, you just see this wide variability amongst species and how they respond to changes in their environment. So what do we not know about corallines? We know quite a bit, but we still have a lot of information that is lacking. So prior to my work with my supervisors, there was actually a lot of molecular information that was missing. We had some partial genomes, but no transcriptomes, which I'll get to momentarily. But anyways, molecular information was lacking. There is no work into molecular level responses. So what's happening at the cellular level? What's happening with gene expression? Are things being upregulated, downregulated in response to environmental change? We also didn't know plasticity across life history stages. So some studies had done just the offspring or young individuals, and most studies just looked at adults. So we were interested in looking at what happens across or multi-generationally. And then unsure of what happens or the role of environmental history. That includes evolutionary history, acclimatization history, and acclimation history. So that's what my PhD set out to do, was really provide this missing information that can then help people in the future, especially people looking at coral reef restoration, and how we can provide and predict how coralline algae are going to respond to future ocean conditions. So the first part of my PhD was to really establish this molecular information in corallines. So genomics approaches to answer ecological questions have been gaining speed. We're starting to really use those to get insight into the regulation of cellular processes, the mechanisms behind these phenotypic measurements that we've done in the past, and the pathways as well. And although these approaches are gaining speed, they're still really limited. So this is a table from my first publication of my PhD, looking at whole genomes in different species or different phyla, and then also looking at assembled transcriptomes. So assembled transcriptomes being all the RNA that are within a given sample, so tissue, cell, organism, and genomes being all of that DNA. So if you look at these phyla at the top, so chordates and arthropods and vascular plants, you see a lot of whole genomes and a lot of assembled transcriptomes. And then as you start getting down to algae, you see fewer. And then in 2019, there were 0 whole genomes. There are partial genomes, like class of genomes and stuff for, or chloroplast genomes for coralline algae, but not actual whole organism genome. And there were 0 transcriptomes. But after our work, there are now 4 assembled transcriptomes that are published and 2 more that are unpublished, but on the way. Right. So what did we find, or how did we get this? So I wanted to build really comprehensive transcriptomes, and I wanted to use these transcriptomes in the future for all my other work, which meant that I needed to actually go and do a study and expose different species of coralline algae that I wanted to use in future measurements to all different kinds of environmental parameters, and then take their tissue and actually extract the RNA and build these reference transcriptomes. So I started with 4 species. This really beautiful one is my favorite. That's sporolithin. That's the really old species, we say. Lithothamnion is usually a fan favorite because it's really pretty. Porolithin right here is one of the major reef contributing species. It's probably inarguably like the most important species to the Great Barrier Reef based on its process of reef building. And then Lithophyllum. So they all span, or they all have different clades and span different evolutionary histories. Right. So I did this experiment, and I exposed them all to different things. I collected them straight from the field. I let them in common garden for 2 months. I exposed them to temperature and pH, and then brought all my samples home and started doing molecular work and building my transcriptomes. So because no one had ever done anything RNA related with coralline algae, I probably spent a good portion of my PhD just figuring out how to extract RNA successfully and get a good quantity and yield of my RNA. This slide gives everyone heaps of anxiety, although it's actually quite like toned down from what I actually did. So if you do have any questions after, I'm very happy to go into it. But this is my bioinformatic pathway, basically. So first of all, I'm going to fall over the stool. I extracted RNA. So if you remember from this, so corallines are notorious for having heaps of stuff growing on them and growing in them. So you have to be really careful when you extract the RNA, so you're not getting like random other algae or different kinds of snails, et cetera, growing on them. So you scrape only that colored layer, which is the living layer of the algae. So extracted RNA from the living layer, came up with a whole protocol for that. Sent that off for sequencing at Macrogen. You get these raw read files and basically go through all these different parts of the pipeline to do your de novo assembly of your transcriptome using Trinity. Then you have a transcriptome, and then you work with it more. You annotate it with BLAST, which is a public database. You can annotate proteins, you can annotate different transcripts to actually figure out what is within your transcriptome. So then later when you do future studies, you can figure out what's going on and relate that back. And last but not least, everyone's favorite, R. So a lot of the work I do is in R, especially with transcriptomics and omics analyses. So I will come back to this later to scare you all more. So that was my basic bioinformatic method to actually build up my transcriptomes. And what did we find? So originally we were like, it's not going to be that interesting. We're just going to use these as reference transcriptomes and not actually work to publish it. But it turned out to be really interesting. So we found, obviously, because no one had ever done this, we found heaps of novel genes. Everything was technically novel. But we found things like zinc finger domain sequences, which could be transcription factors, which are really, really important for development. And we found all these different candidates for different processes that made these group of algae, the Coralline algae, the calcified algae, different from other red algal species. So one of the big parts of my work was to actually go back and compare my corallines to published transcriptomes for fleshy seaweeds and see how they're different. So we found that they are different, obviously. They had genes and families of genes that were enriched to be related to things like regulation of transport, supramolecular fiber organization, adhesion, and even things like calcification, which was really exciting for me. So we did find they're really different, and this study actually provided some insight into the evolution of corallines, because we looked at gene duplication events that probably took place in these species, and actually more broadly, probably the evolution of rhodophytes or red algae in general. So it's really exciting. And then I was able to actually go on and do future studies using these transcriptomes, so they were not wasted. So next was looking at actual molecular response of different species of corallines. So I wanted to use those reference transcriptomes, like I said, to actually then look at what's happening at the gene level, what's being expressed, what's being upregulated, downregulated, et cetera. So we can decipher mechanisms underlying species response to environmental change, looking at their transcriptomic response or gene expression response. Environmental stress can obviously influence these modifications, so causing genes to be upregulated or downregulated, for instance, and this can really be indicative of an organism's stress response and potentially acclimation or even adaptation. So you can see within changes in gene expression or transcriptomic response, you can see sensitivity and potentially resistance or infer those from your results. And then a big thing is being able to link your work to actual physiological measurements. So that's what I set out to do. So we know in corallines, if you apply an environmental stress, like temperature or acidification, we know from previous studies that there have been changes in abundance and survival. You get increases in bleaching. So corallines do bleach, not like corals. They're not expelling their zooxanthellae, but they do get bleaching, and that's indicative of mortality. You see changes in metabolic processes, like that graph I showed earlier, and also decreases in calcification. Did I go too far? Okay, so what I want to do, again, multifactorial experiment. I wanted to look at different levels of pH, so low in control or CO2, ambient and high, and different levels of temperature. So the ambient control temperature at Lizard Island, which is where I did my work for 4 months, was 27, nice and warm, and then an increase of around 2.5, so 29.5. And I used 2 different species, Sporolithin durum and Porolithin, should have italicized those, excuse me. And here are their divergence times, and I had 5 replicates of each one. So this is where I did my research. And here's me using very advanced technology, as in a chisel and a hammer, to chisel off CCA off the reef. No corals were harmed, actually. Sometimes. All right, so the first part of this was after collecting them, I brought them back and I basically exposed them to constant temperature and pH for 4 months on the island, and just regulated or maintained them throughout that time. And after 4 months, I measured photosynthesis and respiration. So inside each one of these chambers is the little algae. It's a very advanced setup. So you have an oxygen optode here that's actually measuring % oxygen in the seawater, and then a temperature as well. So I looked at photosynthesis and respiration in both sporolithin and porolithin, because I wanted to actually see if we see a change in these physiological processes or normal phenotypic measurements, if I can link that to my transcriptomic response. Here's the slide that gives everyone anxiety again. So after measuring photosynthesis and respiration, I took individuals and I extracted their RNA. Similarly, just taking a razor, scraping off their living layer. I've sent them off for sequencing. Well, I created libraries and sent them off for sequencing. And the only difference, well, a lot of the other steps I didn't do because I didn't have to, but so once you get these raw sequence files from the sequencing facility, you can actually look at differential expression. So we used a pipeline called CellSeq, which is traditionally for single cell transcriptomics. But since with CCA you don't get a really big yield, it actually worked really, really well for us. And it's cheaper when you get to sequencing. So with CellSeq, this is the pipeline that I did in our external cluster. And then this is where my reference transcriptomes came in handy. So here you have to actually map the reads back to compare your new transcriptomes against this reference transcriptomes to see what's happening, the regulation of different genes. And then you create a counts matrix, export it to R, and you can get differential expression data. So this is what I looked at, and you get these massive files that say, you have a false discovery rate of all these genes, and if they're below 0.05, we consider that a significant differential expression of that gene. And then you can go and take that into BLAST and other databases to actually identify those genes if they're not identified from your original transcriptome. And then you can look at enrichment and all these other different bioinformatic tools to understand really what's going on, and to then be able to relate that back to your photosynthesis and respiration results. So if you look at oxygen produced, photosynthesis versus oxygen consumed, respiration of my 2 different species. So this is sporolithin here, and top part of the graph being photosynthesis, and bottom is respiration. Sorry, the axes are not aligned, but you'll live. And this is oxygen, micromole oxygen per surface area of the algae per hour. And here we have different pH treatments at the bottom, as well as temperature. So if you look at sporolithin, there was no significant difference in either photosynthesis or respiration. So you see a little bit of a decrease, but ultimately wasn't significant. And then if you look at porolithin, which is this one here, you'll see changes in photosynthesis, not respiration, but you do see a significant change between the control of 27.2 at reduced pH and the elevated temperature at reduced pH. So that was quite interesting. Wasn't totally unexpected because CCA do respond differently to warming and acidification, and we know that because effect seems to be species specific, as does magnitude. But then I looked at my differential expression analysis and looked at differentially expressed genes, and we found something super crazy and super exciting and very, very, very unexpected. So both species exposed to 4 months in temperature and pH treatments, And what we found was that sporolithin had absolutely 0 differentially expressed genes. Blew me out of the water now, so I understand. And porolithin had over 400 differentially expressed genes. So again, super unexpected, but quite cool. So now we know that if you look at this phenotypic measurement of photosynthesis and respiration, that there's no real change, no significant response. And then if you actually look at what's happening with their transcriptomic response, again, 0 differentially expressed genes that are significant upregulated or downregulated, whereas porolithin was quite reactive. So we are terming that as a stable transcriptomic response and a label for response to viral stress. So then if we look only at porolithin, we look at a heat map looking at differential expression between the elevated temperature and CO2. You see that this really nice clustering of genes. So these are a comparison with VST stable, variant stabilized transcripts. So you can see this cluster here of upregulated genes or transcripts, and this is only in the elevated temperature and elevated CO2 or reduced pH, so only in the combined stressful treatment. And then you see this, again, cluster of nicely downregulated. So these were the actual 2 clusters that R came out with. It wasn't me just putting those clusters in. This is statistics. And then you can see kind of a mix of all the other treatments. Maybe there's some clustering with the ambient, so AA being the control, and then here's just elevated temperature and elevated CO2 or reduced pH. So no real clustering amongst these other treatments. So it's really quite interesting because you see this really pronounced effect in the combined stressor treatment. So then we really wanted to link the transcriptomic response, the differential expression of genes, to what is happening cellularly. So we came up with a nice little model, and we found that 51 of the 130 upregulated transcripts encoded for enzymes, and this is the 130 that are in the combined stressor, not the total 190, just to clarify. So 51 actually encoded enzymes. That's really exciting. We can put that in a cell and kind of conceptualize and map out what's potentially happening. And we can link this to changes in crucial metabolic processes that are actually impacted by the synergistic effects of ocean warming and acidification. And I'm happy to spend more time on this later, but I know that I'm probably talking too much, so I won't do that. But each differentially expressed gene, so blue is for downregulated, red is for upregulated. This is the chloroplast. So that's really what we focused on happening, obviously photosynthesis and some membrane proteins as well. So this part in orange is the Calvin cycle. So you can see it's differentially expressed depending on which gene. If you look at glycolysis, it's actually all upregulated, or the genes that we found to be involved in that process are all upregulated. Whereas we found with the non-oxidative branch of the pentose phosphate pathway, these are actually all downregulated, and some differential expression here in the oxidative. So again, happy to spend more time on this, but we'll shoot through it now. But that allowed us to actually link physiologically for our photosynthesis and respiration measurements, kind of try to connect the two between the expression of genes and these processes. So then this is actually the first part of my PhD, but I presented later. So I wanted to figure out what's happening across life history stages. So I wanted to be able to compare adults to their offspring or germlings for coralline algae. The experiment actually started as a transgenerational experiment, but then we realized that this species of algae, it grows extremely slowly, and that did not work. So you have to be able to be adaptable in your PhD and realize that probably not everything is going to go your way. So that's what I did. Cut that experiment after 7 months of running it, and then looked just across different life history stages. So we know the environment can influence the phenotype, so we can see changes in respiration and growth and photosynthesis, what happens to the offspring of these adults that are already experiencing stress. And then also with the different life history stages of corallines, it's really interesting because adults are calcified. Totally, they calcify within their cell walls. But when they actually shoot out their offspring, I'll show a picture later, those are totally uncalcified. So the spores, when they first come out, are uncalcified. So they're quite a cool model to actually look at calcification if you compare adults to spores. So I wanted to look at, again, multifactorial, look at CO2 or pH rather, and temperature and how those impacted adults, and as well as the germlings or the offspring of these different individuals. Fanny will commiserate with me because I built an aquarium room that was not on the ocean. And it had a lot of wires and it was very precarious, but I did it and it ran for 7 months without too many hiccups. I had about 60 different tanks. So in each one of these tanks had an adult at first. And then after about 2 months, I induced, aka stressed the adults out quite a bit, and they started taking out spores. And that's how I got my F0 generation. So that's how I have babies, and I put them, let's see, I did a picture. So these are the adults. I induced release of the spores, and this was actually after like 2 months of them being in there. But you can see all these tiny little red dots, and those are the germlings. So this is actually when they've calcified now. And I picked probably about, I don't remember how many individuals, and I tracked growth and survival on them for a good portion of time. And so this, I kept running for almost 7 months, I think. So at the end, what we found was adults for sporolithin, shockingly, were extremely robust. So this is the same species that we saw 0 differentially expressed genes after 4 months in treatment as well. So we found that they somewhat reduced photosynthesis, not significant. There was no change in survival. Literally every single individual that I put in at the beginning of 7 months survived, no mortality, no bleaching. They're hardy buggers, and no change in respiration. Whereas germlings, we saw a significant decrease in survival and in growth, and this was primarily attributed to the combined effects of warming and acidification, but temperature was a major player when you looked at those models. So then I wanted to really explore development at a transcriptomic level, and I haven't presented this data before, but I thought it was pretty cool to share, and also gives you an understanding of calcified versus uncalcified structures. So these come out like little noodles. So this is the adult, and these are the spores. So a lot of corallines tend to release when temperatures are higher. Makes sense, lots of animals do that. Organisms do that, rather. But these are all the spores. They come out in packets of 4. We call them tetraspores, conveniently. So these are all uncalcified. They only stay uncalcified for not very long until they actually settle, and then they start binding more and calcifying onto or cementing onto a substrate. And they're negatively buoyant, which is quite convenient for research purposes. So a lot of times they'll start to settle on the adults, or they'll just kind of drop to the side. They are not like planar larvae or something swimming around. So they're quite easy to control. So yeah, these are the uncalcified. So for transcriptomic purposes, because I wanted to look at development and differences at the transcriptomic level between adults and their spores, I actually took just one of these little guys, and I did transcriptomics for 3 different species, and I will share 2 now because I don't have the results for the other one yet. So this is for the species porolithin, the one that had over 400 differentially expressed genes in response to environmental change, and well, I found a lot. So if you look at a false discovery rate, which is equivalent to saying P is less than 0.05, we found over 6,000 differentially expressed genes. About 2,700 of these were downregulated, 34 upregulated. So this is comparing spores against their ambient adults. And again, very cool. We get this nice clustering here. So here are all the spores that I did and the adults. So you can see all these genes are upregulated in adults that are not upregulated in the spores, and vice versa. So we're really looking for things like homeobox proteins that are really important for development and differentiation, or sequences similar to that, because again, Coralline algae database is completely missing for, doesn't have high representation for algae in any of like the non-redundant databases or when you look at proteins. So we're kind of doing a lot of similarity searching and phylogenetic analyses for these different transcripts. And then also, everyone's super interested in carbonic anhydrase when you think about calcification. And especially with corallines, everyone's kind of had this idea that it plays a role, but no one's actually known what exactly it does or if it's even present. And it is present, or a similar sequence is present. And we see that in the 2 different clusters. And then also homeobox proteins, like I said, very important potentially for development. And they have been found with plants as well to play a role in development there. And this is sporolithin. So the adult that showed 0 differentially expressed genes, it's not just because my data was wrong, and it's not just because I did something incorrectly, that just didn't respond to the treatment. So when you actually look at adults and spores, you can see, again, lots of differentially expressed genes. So an FDR of 0.05, you get almost 20,000 differentially expressed genes. If you lower that down, sorry, I didn't talk about that last time, you still get 14,000 differentially expressed genes. So they are different and things are happening. It's not like they just don't respond at all, or you can't compare them. So again, you see this really nice clustering of the adults and their upregulated genes here, or transcripts, and then the spores, again, clustering here, showing differences in what you'll have switched on and off, or upregulated or downregulated. So this work is still very much in prep, but so far we're finding pretty exciting stuff. Again, we're seeing like opposite response and transcription factors, and we're trying to now pinpoint. Transcriptomics is extremely exploratory. You get a lot of data and a lot of stuff, and you kind of have to weed through it all and figure out what your question is, and hope you stick to it, even though you find really exciting things along the way and you get very distracted. So I'm working on this now. Hopefully we'll be able to tell a really cool story about the development. And I have one more species that's being sequenced now. So hopefully we'll be able to tell this cool story about development in corallines, which hasn't been done, and which will help us, again, with understanding how they'll respond in the future. So the last part of my PhD, and I have no idea how long I've been talking, sorry if it's been too long, was looking at environmental history, acclimation, and acclimatization history, and this was purely looking at photosynthesis. So I left molecular work for a little bit to just focus on traditional physiological measurements. So we know that with CO2 increasing in the atmosphere, you get an increase in heat waves as well, and also these acute temperature events where you get sudden shocks of temperature. And it's possible that acclimatization history, so that they experience in the lab, or that you manipulate, and acclimation, so that which is in the environment, plays a role on thermal limits. So those being those physiological limits that all organisms have, this nice little curve or thermal tolerance range. So organisms generally function within this range. Surpassing it on either side is very bad, death. And then they have this optimal range with temperature and their performance, whether it be photosynthesis or running or something like that. And I was really interested, based on my previous work, if populations, since you have this really wide distribution of corallines, you get different populations of the same species across different reefs. And I was interested in whether these populations will respond similarly, if their environmental history plays a role in how they respond, and if different populations may be living closer to their optimal performance or their higher end of their thermal tolerance range, or farther away from this range. So this was a two-parter. Again, love manipulating CO2 and temperature. I was up at Lizard Island again, and I used only porolithin because this is a species that most people are really interested in when you think about reef structure and stability, because they're like the major reef contributor, and that's reefs worldwide, not just the Great Barrier Reef. So manipulated things, kept them in treatment for about 8 weeks, 2 months, and then measured photosynthesis whilst increasing temperature by 1 degree per hour until reaching about 33 degrees, which is an ecologically relevant change or increase in temperature that you could see within 1 day on the reef crest of the Great Barrier Reef. So I collected these specifically from the reef crest environment. So if you look at a temperature in 1 day based on a hobo logger, you can see an increase of like 5 degrees in a day. It's pretty crazy. So continuously measured photosynthesis as I increased temperature by 1 degree per hour. And then the second part of this work was to compare different populations, and this was without manipulating any environmental variables. So without changing temperature or anything, I just went to each one of these locations. I went and collected, left them to acclimate for about 2 weeks, and then went and did these ramping experiments with about 24 individuals, which took quite some time. And again, measured percent oxygen in the water over time, and took these individuals, instead of just going to 33 degrees, we tried to take them until they kind of reach this upper thermal limit, or what we coined as an upper thermal limit for these algae. So when respiration takes over photosynthesis, and you start seeing a decrease in the percent oxygen faster than your increase. So I compared these 3 different populations. So if you look at acclimatization history, so that history that they experience in the lab, so what I manipulated, there's a lot of variability, but you can see the elevated temperature decreased the tolerance to acute thermal stress. So we didn't call it a heat wave, we called it acute thermal stress because it was a fast change. And based on linear models, we found that temperature is the driving factor for actually this significant change in photosynthesis, but we did also find that there was a decrease in photosynthesis no matter what, which kind of hinted at that potentially this species or this population of porolithin is living closer to their thermal limit now. And this was further supported by the population study we did, and this was a pretty cool turnout, I think. So we've then wanted to look at acclimation history, so environmental history, and how that may play a role, and look at these different populations, see which one's living closer or farther away, or if that's even a thing for corallines. So we wanted to compare existing tolerance or living at or near their thermal limit. So this is the lizard island population, the one I just showed for the temperature and pH experiment. And even after 2 degrees, you see this like crazy drop in photosynthesis, and they kind of plateau out and then keep decreasing. If you look at the most southern population, so Lord Howe Island is the most southern true reef in the world. It's like pretty far down, and the water is much, much colder. Not like here, but still very cold comparatively. So if you increase temperature every 0.5 degree, you see a decrease in photosynthesis, which was interesting. And then Heron Island probably was the most shocking result. So this is a population that's in between the two, almost exactly, about 2,300 kilometers to Lizard and to Lord Howe. And they actually maintain their level of photosynthesis or oxygen produced, or average oxygen produced, up to about 31.5 degrees. And then I ran some tests here to look at like the actual breakpoint. Statistically, we found that Lizard Island was had this breakpoint a little under 30 degrees, and Heron Island didn't have one. I stuck it in the graph because otherwise it looked weird. But they did not have a statistically significant point of temperature at which they broke. So when they started respiring more, or consuming oxygen, and then Lord Howe was the lowest. Obviously, you see this change in photosynthesis, and it's continuously decreasing. So this paper has been prepped with my supervisor for 2 months. And conclusions to everything. So my thesis ended up being like quite bits and pieces, so I had to really tie it together in the end. But we found really that CCA are different from other red algal species, fleshy red algal species, at a molecular level. And the work we did really led us to explore the evolution of corallines in general. We found and supported previous findings that response and magnitude of response is species and life history specific. So depending on what species you're looking at, they'll respond differently, and also across life history stages, you're going to see something different. So earlier life history stages were quite sensitive, whereas adults were more robust for one species. Transcriptomic analysis can reveal sensitivity and resistance. So from our work, we inferred that sporolithin, that really hardy species, is potentially more resistant to changes in its environment, whereas porolithin was found to have transcriptomic lability and potentially is more sensitive and lacks the potential for acclimation to future oceans. And then we provided insight into the development and calcification. I'm also doing other work looking into calcification with these guys. And then evolutionary history could be influential. However, that definitely needs to be systematically explored because I only did 2 species. So it'd be very cool to broaden that and do a bunch of different species across the phylogenetic tree and actually look at that idea if it holds, if older species are less or more robust than these more speciose species. And then environmental history of these corallines does play a role, depending on where they are or what they experience, that can impact how they respond and the magnitude of that response. All right, that's it. Thank you to everyone from past labs, etc., and for you guys for being here and listening to me blabber on. And I'll take questions.
Merci. Thank you, Tessa. So we have a question online de Geneviève Parent.
Salut Tessa, hi. Welcome, and I'm really glad to see all of your expertise, and I'm flabbergasted by the amount of work that you did during your PhD. So it's really nice to have someone working on transcriptomic in the region too. So welcome. And it was hard for me to understand all of the details of what you were presenting, but I understand that you were making it also accessible to biologists in general. So I have more questions and details to ask you on Friday, but for today, I'll keep to some more simpler question. So in your last experiment, I was wondering, if you go back to the slide where we're showing some critical point where the 3 population have different answers in photosynthesis. I was surprised also that you did not have an intermediate response, let's say for Iron Island population. And I guess this is what was surprising you too, why is that population? So it led me to some results we got for other species, so it's for shrimps also, where it's not the actual environmental condition that you may be seeing as adults are living, but really those where the organism were living through its development that are highly influential. And maybe have you looked at the conditions during the earlier phase of the organism? Are those intermediate for the Iron Island environment compared to lizard and Lord Howe Island? Or have you looked at other parameters
where they
that could
are
explain?
in development?
Yeah, exactly. Or during their development to explain a ontogenic effect or maybe a maternal effect or something like that.
I mean, just ID'd them to being adults, so you can tell pretty easily with corallines, once they actually start having reproductive structures, and then that hints that they're adults. We kept all the fragments around the same size. Sorry, I should have included that. They're all around 3 centimeters squared. So they were still around roughly the same size. And then also I made sure, because I think it's really important, also we've seen it in other work, to make sure that they were the same phase. So rhodophytes and corallines have asexual and sexual reproduction. So almost all of the individuals I worked with were always an asexual. So you can tell that based on the reproductive structures. So with this species, you can tell if they have a conceptacle, and within that conceptacle, you have to like poke them and see what comes out. And as long as the spores, so the babies come out in tetraspores, that means that they're in their asexual phase. Whereas if they're sexual, you would see these things called carpal sporangia, which are these like, they look different from tetraspores. And technically what's supposed to happen is that one fragment of algae should only be in one sexual phase. I do not think that's always true, but that's the leading understanding of phycologists, is that you can't have one fragment be both asexual and sexual, but you do have this phenomenon called coalescing, where different individuals can like grow into each other when they're young. So it's possible that you can get both life history stages, but phycologists would disagree with me for the most part, I think. But yeah, that's all you can really do, is just know the size and also what phase they are in. But it's like there are definitely potential for confounding factors there that might have, caused the response. And for Lord Howe, at that the year we went, the air surface temperature, or the air temperature, was actually about 1.5 degrees higher than it is normally. So that could have been providing additional stress onto this population, because I didn't think it was going to respond quite so significantly decrease, but it did. So it could be that they were already stressed out. We actually saw like a major bleaching event whilst we were down there for a month. So it is possible that they were already experiencing some stress, and that could have impacted the results.
Thanks. And if I can, I have another short question. Okay. Like, one interesting result you got is that there was no differentially expressed gene for the temperature experiment for sporoliton, where you did have some for Coralline. I was just wondering about like how you did your reference transcriptome. Like, what did you include to make it? Like, did you include individual that did live at two different temperature, and like just some?
The reference transcriptome had like as much as I could possibly get. So then anyone in the future that wants to work with CCA, that want to manipulate temperature or look at different reproductive stages and things like that, I used all of that, pooled it together, and built the transcriptome. And then I still had my control treatments, and then my perturbation treatments as well, to then map back to that and compare.
Quite surprising.
Yes, very. And I actually like reanalyzed that. So I used a DEC first, and I was like, there's no way this is possible. And then used EdgeR and got exactly the same, like, well, more or less the same results, and not significantly different. And then if you look at the heat map for FDR above 0.05, you do see expression. It's not like they're just innate, like not responding at all, or that something went wrong. So you do see like expression, it's just not significant.
Thanks.
So I had two questions. Sequential, you, in the first phase of your presentation, you talk about synergistic effects.
Yeah.
And I was wondering whether are synergistic or you meant combined?
Combined.
Okay, and then the second question, do you see evidence for dominance or temperature effect in your responses?
Yeah.
Like between the combined treatments and the temperature?
Yeah. Okay. Yeah, so generally when I did like my statistical tests, and did post hoc analysis, it was always the temperature that seemed to be driving the changes, or what we found. Yeah, so yeah, temperature.
So the effect of acidification are only subtle or opposite?
Sometimes they, generally quite subtle, but they would enhance the effect of elevated temperature when combined. But if you looked at just pH, we saw like with sporolithin, they kind of suppressed photosynthesis with increased or decreased pH, which in land plants can mean like plasticity or acclimation. So in land plant studies, they found that if they reduce photosynthesis in response to elevated CO2, it's actually indicative of like a acclimatory process, which is quite interesting. So another reason we inferred that this species was more robust and had the ability to acclimatize or acclimate.
In a few words, could you explain us what is your postdoc project?
How many words? Okay, I better get this right. So I am working to integrate different omics analyses. So I'm going to integrate data sets and looking at transcriptomics, hopefully, but primarily metabolomics, lipidomics, and proteomics. And what ideally we want to do is be able to integrate these data sets and use them to like predict what's going to happen to organisms in response to environmental change. And ideally we'll eventually be able to kind of create like a network and see what happens if you look at this response data set. So say this change in photosynthesis or in the metabolome, what's going to happen in different other compartments of that organism. So what's happening in the lipidome or the transcriptome or the proteome. So right now it's really working with existing data sets that are free and not being used for proteomics and metabolomics and stuff, and reanalyzing those and then trying to integrate them.
Okay, thank you very much.
Do you expect to observe the same processes for cold water corals?
I'm not sure. Yeah, that's a good question. For corallines, potentially, yeah. I'm not incredibly sure. Like, so you get similar species. Not exact species, but congenerics across like a huge range of areas, especially with sporolithin. They can be temperate, so living in Tasmania, which is our cold water in Australia. You have to wear a dry suit there. I think it gets down to 10 degrees. But you see they do respond. And you can see a response even after like, well, yeah, they're more muted response in comparison to other coralline species. But yeah, I'm not sure about cold water, but potentially. I mean, temperature would definitely probably play a role here. Yeah, it'd be fun to do. Let's do it
next part.
Yeah. I do love algae.
Okay, so thank you so much for presenting your PhD research and future research here. So I let you go, I think. Merci à tous d'avoir assisté à notre webinaire. Je vous retrouve la semaine prochaine pour notre prochain webinaire avec Yann Rick qui viendra de nous parler des tourbillons. Donc à la semaine prochaine dans la même salle et en ligne. Merci.